तू इत्थे रखा करना मैं
स्टार्ट होगी मेरी क्लास। पैसे दे दो खुले है। Very good evening, class. How are you all? I hope you are doing good. A very warm welcome to today's sessions, day two, where we will go through a lot of exciting things, a lot of different things also. And yeah, how are you all? How was yesterday? Did you spend some good time with family or with your friends or roam around or stay at home, stay safe? How was it? I just grabbed some jalebis. That's a common ritual that we do on the Shara, and we just brought it and we ate with my mother, father, sister, and then yeah, that's it. Not much. So as soon as it's 6 o'clock, we will start our lectures. Do you have any questions so far? Anything else you want to talk about? Or we just start with our lectures today? Good evening, Juby. Good evening, every all students. So, so I hope most of the students have joined so far. Let's wait for another 1 minute, then we start our session then. So mainly students, these lectures that I'm teaching you, they will be mainly subjective paper like lecture type. I have taught them already at the university level. So these are really, what I'm teaching you is from the university level syllabus, and the lectures are also guided by from the same. So it's all pure university based Indian Agriculture Research Institute, AIRI. So from them, these lectures have been guided out. So they are more focused from the plant biotechnology and like more on the plant side of genetics. So we will see how numerical aberrations and chromosomal aberrations of chromosomes, how they are concerned with the our further lectures. So we will discuss them in more details in the last lectures we will go through. So uh, Any questions regarding any assignments or review? All things are clear yesterday. So I took a couple of much time of yours yesterday to just describe everything so that we can have a good start from yesterday and we can start in a good way for today also. So all good from yesterday, all things are clear. Okay, nice. Please students interact with me. That's what these lectures are meant for. Please write whatever you have any questions or want to discuss something about project or anything. Feel free to discuss here. So I will be very happy to interact with the students, not just going through the slides and giving the lectures and letting it off. So I will be happy. We are interacting with each other's. Good evening, Bache. Very good evening. A very warm welcome. So, it's been 5 minutes now. So, I think we can start with our lecture of today's, that is Principle of Genetics, Mendelian's Law of Inheritance. So as were discussing about concept of inheritance yesterday, so we discussed about various principles, various history about it, from Lamarckism, Darwinism, Anton von Leeuwenhoek, and germplasm theory. Then we discussed about many other theories, like so many theories yesterday, about brief history about them. Then came the father of genetics, our Mendelian law, who brought 7 traits about dominant and recessive, and we discussed about various characteristics, how he brought his theory of genetics and bringing thing called factors that could be the reason for the inheritance that was passing from one generation to another. So that's what we did yesterday. So let's start with today's lecture about more detail about Mendelian's law of inheritance. There were 3 laws basically he gave: law of independent assortment, law of dominance, and law of independent assortment, law of dominance, and law of there's one law more. I lost it. Law of segregation. Sorry for that. Law of dominance, law of independent assortment, law of segregations, and there are also some exceptions to that. Law of codominance, like law of lethality. So we will go through them also, and basically we will discuss about how test cross is done, how various hybrid ratio, and how a single cross, dihybrid cross, trihybrid cross, they're all done in this case. So we will go through each part of this lecture, and I will always as always discussing with the students that our concept of today's lecture is that we will deal with your this whole syllabus of yours, which is divided into unit 1 and unit 2, and this takes us to complete almost one semester, six months. But we will try to manage to finish this in 10 days and able to finish your 1 6 months 1 semester course um, into your in this 10 days workshop. So this is quite challenging for me, and for quite challenging for you also, for all the students, to grab everything within this short span of time. So let's start with first law, that is Mendelian's law. So first a little bit about introduction about it. So So Mendelian first discovered, uh, yeah, discovered the crossing white and purple flower. So he crossed white flower and purple flower, and the result was not a hybrid offspring. So there was no hybrid came out at for what he did first. Rather than being a mix of two offsprings was purple. So when he mixed purple flower and white flower, so the mixing of it, so he removed stamens from the purple flower, and from the white, uh, he took the stamens of the white, and then they mix transform the pollen from the stamens of white flower to the carpel of the purple flower. Then they pollinate them to mature into pod, then leading to the planted seeds from the pods further. Yeah. Then we have examined offsprings all purple flowers. So first generation F1 generation came out to be all purple. So he conceived the idea that whatever the heredity that was passing from one generation to generation, another generation, that thing is called factors. And one which is recessive character and other is known as dominant character. So there are two factors that they were playing role, that is recessive character and dominant character. So Mendel said that factors, later called genes, normally occur in pairs in ordinary body, yet segregate during the formation of sex cells. So each member of the pair becomes part of the separate sex cells. So in this case, what was happening? The purple flower was dominant, white color was recessive. So purple color was taking the hiding the recessive gene of white flower. So that's why everything was purple in this case. And after the Mendel self-fertilized F1 generation, so he first generation was self-fertilized, he obtained the ratio of 3 is to 1. And then the ratio he divided into three different parts, that is capital A, small a, capital A, small a. So capital A represent the dominant factor and small a represent the recessive character. So that's how he brought the dominant and recessive characters into the F1 generation, who was F1 who was self-hybridized, and then he brought this recessive, dominant, and this was a hybrid one. So initially, what happened in this field of Mendelian's law? Mendelian stated that each individual has two factors for each trait, one from each parent. So each factors that were coming from the two factors of each trait, from one from the parents, one from mother, one from father, and the two factors may or may not contain the same information. They could be the same or either they are not same. If the two factors are identical, the individual is called homozygous, like A, capital A, small a. If they are different, so homozygous will be capital A, small a, small a. If they are different, like A, capital A, and small a, they will be heterozygous. So the alternative form of a factor called alleles, the alternative form then of factors are called alleles, and the genotype of an individual is made up of many alleles it possesses. So an individual physical appearance or phenotype is determined by its alleles as well as its by environment. So an individual possesses two alleles for each trait. One allele is given by the female parent and other by the male parent. So as we discussed already, one from the male and another from the female, and they pass as an individual matters they're producing. From female we have eggs and from the male we have sperms. So that was some traits that he was bringing out by Mendelian's law. And further, in the heterozygous individuals, only allele that is expressed is dominant. So in heterozygous, like capital S, small a, so mainly the capital A, the one with the dominant gene, will be expressing that. Recessive allele is still will be present, but it is hidden. So this will be hidden one, not present like big one. Okay. So let's start with the various laws now. So he postulated three laws, which is known as Mendelian's laws of heredity, and these are the law of dominance and recessive, law of segregations, and law of independent assortment. So first, let's start with the law of dominance. You might have done that in your plus two class many times, but before, if we are starting with something, some basics of genetics. So core subject name is principle of genetics. So we will be dealing with the all genre that is touching the genetics part. So we are going through its first principle, basic principle. Then we touch the higher part, like type of gene actions, qualitative and quantitative traits. So that is something advanced part of it. So we will go gradually with that. So at the moment we go through the basics part. So when two homozygous individuals with one or more set of contrasting characters are crossed, the characters appear in F1 hybrid were dominant character, and those do not appear in F1 are recessive characters. So like this. So here we can see there is a um, capital B capital. So we have this female from the pistil, capital B and small b, and capital B small b. So whenever we make a cross, so this is a Punnett square method. So we put capital B small b like this, capital B small b like this, and we take one of their alleles like in the cross. So first one, capital B, capital B, capital B small b, capital B small b, small b, small b. So because capital B was a dominant character, so purple color, so we always see purple color in this case, purple, purple, purple. But when the both recessive genes were present, we saw a white color. That's why we saw 3:1 ratio in the F1 hybrid. So dominant and recessive of the genes can be explained on the basis of enzymatic functions of the genes. Dominant genes are capable of synthesizing active polypeptides or proteins that form functional genes. So why this is happening, that mainly the dominant genes is coming out, it's happening because of the active polypeptides or proteins that are forming functional enzymes. So they are producing functional enzymes, where the recessive genes code for incomplete or non-functional peptides, whereas in your recessive genes here, they are going through the incomplete or non-functional polypeptides. Oops. Non-functional polypeptides. So therefore the dominant gene produce specific phenotypes while the recessive gene fail to do so. That's why we see always purple color, because of the active polypeptide chain in them. Then in the heterozygous conditions, also the dominant gene is expressed itself, so that the heterozygous homozygous individuals have similar phenotype. So in the case of law of dominance, if there are two alleles coding for the same trait, if there are two genes coding for the same traits, one is dominant, it will show up of the organ, while others will won't. So the one with the dominant character will show it and other might not. So like in this case, cap small b small b. Okay, so that was something about law of dominance. And what are the some importance of law of dominance? The phenomena of dominance is of practical importance, as well as the harmful uh, recessive characters are masked by the normal dominant characters in the hybrids. So in human beings, form of idiocy, diabetes, hemophilia, these are all genetics related diseases, are recessive characters, and the person hybrid of all these characters appears perfectly normal. If somebody is hybrid character like capital A small a, they will be perfectly normal, but harmful recessive genes can exist for several generations without expressing themselves. So these recessive genes could still be present for several generations without harming the persons. But to this law of dominance, there is an exception called incomplete dominance. We will discuss about that also, where we see blending of two characters. So instead of a dominant character being highly expressed there, we see blending of the two parents. So in that case, hybrid were found to be midway between these two parents. So somewhere in between, we got the results, and this is called as your incomplete dominance or blending inheritance. It means that two genes of the allomorphic pair are not related as a dominant recessive, but each of them express itself partially. So they're 50% 50, 50 50, they are like expressing 50 percentage of each part. Okay. For example, Mirabilis jalapa, this is a 4 o'clock plant. When the red flower crossed with the white flower, capital R, small r, hybrid came out to be F1 plant that bears pink flowers. So first thing that came out from the red and white was capital R, small r, that is a hybrid, and it gives a pink color instead of the red colors. And these pink colors are self-pollinated. They develop red, pink and white followed in this ratio 1 is to 2 is to 1 in the F2 generation. In further F2 generation, they produced 1 is to 2 is to 1 ratio. Then comes the law of segregation, the second law, that is the purity of gametes. So this Mendel summarized his findings in two laws: law of segregations and law of independent assortment. Law of segregation and law of independent assortment. So law of segregation states that when an individual produces gametes, so it's all about they're producing individual gametes, and the copies of each gene separates. So if there's R and R, capital R, small r, so we take the one individual of each gametes, and each gamete receives only one copy, and only one copy is being transferred to the another gamete. So that is known as your law of segregation, that is purity of gametes. So law of segregation states that whenever a pair of contrasting factors or genes of allomorphs are brought together in heterozygote form, in the hybrid form, the two members of allelic pair remain together without being contaminated, and when the gametes are formed from the hybrid. So you can see that here in this case, so capital R, they are the genes for the round, capital R, small r, they are genes for the round, uh, for a round, and small r, small r, they are meant for the wrinkle. So this is wrong. This should be small r. So this is how you see the ratio of round versus wrinkle. So this is what the purity of gamete says. So you took one gene from there, one allele from there, and then there's one R and small r, and then there's a cross between them further by the Punnett scale. And what we see is biological generalization. So in the Punnett scale method, we saw the ratio of round into wrinkle 3:1. So this another part of it, another example. So you have purple flower, white flower, you made a cross. So F1 generation is F1 all purple. Then you make self-crossed F1 and F1 crossing with the F1 generation. So F2 generation is again 3 is purple, 3/4 plants have purple colors, 1/4 plant have white flowers. And same goes in here with the genetic makeup alleles here. We have capital P and small p. So we took capital P, and from here we took small p, and then we made a cross according to law of segregation, and you got F1 hybrid as capital P, small p. Then half allele of capital P, half allele of small p, then you made a cross and you have phenotypic ratio as 3:1, 3 purple and 1 white. So it's showing how this on the left, what was showing you, so how actually according to law of segregation these things are happening. And what will be the genotypic ratio? It will be 1 is to, because in genotype we see uh, the allelic formation, like how many of your hybrids are present, so how many of your full dominant are present, how many of your full recessives are present. So that's what we show in the genotypic ratio. So in the genotypic ratio you have capital P, capital P, small p is 2, and small p is 1, whereas the whole 3 purple color, they are 3 and 1 is white. So that is phenotype, that is showing you generalized idea that what you are seeing, the phenotypic structure. and genotypic ratio is telling you the about how many types are present, like that. Then comes the law of independent assortment, also known as inheritance law, which states that alleles of different genes assort independently of one another during gamete formation. So while so that's what independent assortment says, that whatever uh, just a second. So during the gamete formation, the different genes, they sort independently, irrespective of one of each other during the gamete formation. So while Mendel experiments with mixing one trait results in suiting of 3:1 ratio, so whatever we seeing that results were like coming up as 3:1 between dominant recessive phenotype. His experiment with mixing two traits also shows a dihybrid cross with 9:3:3:1. So each of this table show two genes are independently inherited with the ratio of 3:1. So whatever the assortment is going on, whatever the level of difference that what we are seeing was, it was happening according to the it's different genes present. So Mendel concluded different traits are inherited independently. So if the different traits are present, like purple and white, they will be inherited independently of each other. So they will not depend upon each other. So there is no relationship between them. For example, cat color and tail length. So if there is a cat color and tail length, both will be not dependent upon each other. So there is no linking between each other. So they will be, so like if there is a round pea and green color of a pod. So both are different types traits, and they will be inherited independently, and they will be not dependent on each other. So that's what the law of independent assortment says about. So here we have shown you a cross of round and yellow. So two different traits, that there is a round shape and yellow color, and there is a wrinkled shape and yellow color, and oh sorry, green color. So there are two different traits, round yellow and wrinkled green. So you make a F2 hybrid, and in the F2 hybrid you have RY, capital RY, capital R, small y, small r, small y, small r, capital Y. So gametes are producing like this. So you have 9:3:3:1 ratio of round yellow, wrinkle yellow, round green, wrinkle green, like this production. Then law of independent assortment. So let's continue this further. This is to express about the same thing like that we were discussing in the previous diagram. So they have explained this in the subjective manner. So how we came out to be on these results. So in the above experiment, yellow and brown characters are dominant over green and wrinkle characters, which can be represented as follows. So you can see that yellow color is capital Y, green color is small y, round character with capital R, wrinkle character by small r. So therefore the plants with capital Y and small and round cotyledons will have the genotype capital Y small capital R, and those with the green and wrinkled cotyledons will have genotype small y small r. And these plants will produce gametes with gene capital Y capital R and small y and small r respectively. So these are the gametes that you will get. And whenever the cross pollination is done, the union of these gametes in the F1 hybrids, that what we got, capital Y small y, capital R small r. And when these produce gametes, all four gametes have full freedom to assort independently. So these all characteristics of yellow, green, wrinkled and round, they will assort independently. They will be not dependent upon each other. So there will be four combinations, male and female gametes. So like this: capital R, capital R, capital Y, capital R, small r, capital Y, small r, small y. So here in this type. So this shows an excellent example of independent assortment, and these gametes can unite at random, producing in 16 different combinations, presenting four phenotypes in the ratio of 9 is to 3 is to 3 is to 1. So we have mainly four phenotypes, that I have showed you. So you will have these kind of results, and all these results are independently assorted. This has been again explained how it was we came out to the results. So now there are some deviations, some exceptions to the Mendelian law. By now, so far, if we go further, any questions, students, so far? It's I'm trying to make as simple as possible so that we should not have any confusions in later part. All clear? Please, please interact so that I could know that you are present. Everything is good, students? Aha. So when we are speaking, things come out to be later. So your messages coming out to be later. So we need to, yeah, wait for 2 minutes or so. Not 2 minutes, 30 seconds. Maybe there's a buffer of 30 seconds. So when I'm speaking, so it takes around 30 seconds or 40 seconds that our voice reaches to you, and then I'm able to read your messages. And so after speaking, I will wait for 40 seconds so that I'm sure the messages has been, the voice that I was giving the message has been reached to you and you're able to reply. So okay, good, very well. So these are the three Mendelian deviations of exceptions of anomalies that are incomplete dominance that we have discussed just now. Mirabilis gelappa, with the exception of red and white flower producing pink flower, with the ratio of 1 is to 2 is to 1 in the F2 hybrid. So this is incomplete dominance. That was the first thing that came out to be. So it doesn't happen that complete dominance was happening. It was the red flower RR and white flower small r. They were producing first generation as pink flower, and then when we make a crossover of these flowers itself, then we have a red, two pink, and one white. So like this. So this is wrong. So there are sometimes you can identify the errors here itself. So two pink would be capital R and small r, and one red would be capital R, capital R. So this has to be there and this has to be there. So just keep these things unflawed. So this was the mechanisms that how the red flower and white flower was done. So you can see capital R and white flower with the smaller and smaller. If F1 hybrid is capital R, small r, that is pink flowers. Then red, pink, white. So you have red flower, pink flower, white flower. So generation of you have independent, first of all, law of segregation. You have CR and smalls. This is CR and then CW, white, red. So you have one single gametes CR and CW. Then first one is CR-CW. Then you take again half gamete CR-CW, and then you make a cross with the O1 sperm again in the F2 generation. And then this you have one red CR, then CRW two types, 1 is to 2, and CWY. So these are pink color and this is white color. And this is how it is all done. Then comes the codominance. So in case of codominance, both allele express their phenotypes. Both like in the heterozygotes greater than the intermediate one. So the example is AB blood group in human. So like the people who have blood group AB, capital A, capital B are heterozygous exhibiting both capital A and capital B alleles. So both are being expressed. So in other words, heterozygotes are for codominant alleles are phenotypically similar to both parental types. So phenotypically, they will have the characteristics of both parents, capital A and capital B in their genes. So main difference between codominance and incomplete dominance lies in the ways the genes acts. In case of codominance, both alleles are active. So you have both genes are active, while in the case of incomplete dominance, both genes are blend together to intermediate one. So like we have capital T and we have small t. So in the one with the capital and small t, both will be expressing. So here this is an example. So we have these designs on showing to you that this is the phenotype here and this is the phenotype here, and in this both phenotypes will be present and both will be active fully, not partially. But in the incomplete dominance they were partially active, so there was blend of genes actually, mixture of both. So that's the major difference between incomplete dominance and codominance. Okay, now lethality. So here in the lethal genes, so they are present. If the lethal genes are present, most of the time, mouse will die. So in this case, capital Y if present in the case of mice. So that means the mouse will always die. So we have a gametes of yellow color of capital Y small y and capital Y small y. We did the meiosis, so we have gametes capital Y small y, capital Y small y. So we have a fertilization, so F1 generation. So capital Y, capital Y small y, and small y small y. So half of your generations are yellow. One fourth is dominant, but whenever the dominant genes are present, mice has to die. But but 2/3 of a progeny is yellow, and 1/3 of a progeny will be non-yellow, like in this case. So that's what this lethality depend upon your genes or what kind of genes are present. So lethality genes could be dominant, conditional, semi-lethal, or synthetic. So they could be in any form depending upon the genes involved in them. Now comes the monohybrid cross. So this is a cross that is made, and this is a cross made between two breeding parents differ from single traits. So these parents intercross to produce F2 generation. So monohybrid means only one trait that we are seeking. So that's why it's called as monohybrid cross. So homozygous green, homozygous yellow. So it's a pod color that we are seeking. So we take a gamete, capital G, small g. You made a cross, F1 generation is capital G, small g, capital G, small g. Then the F2 generation would be crossing of F1 by self cross, F1 by multiply by F1. So capital G, small g, capital G, small g. So these are the Punnets here. So you got capital G, capital G, capital G, small g, small g, small g. So like you have four different traits. Then this is a dihybrid cross. This is the following legends were described for the piece by Mendel. So you have a tall, two traits that we take in this, and green pod, and dwarf and yellow. So this is how we do it. So pure breeding parents can be crossed to produce hybrid, dihybrid, meaning that two genes affecting different traits are heterozygous. So when F1 is self fertilized or crossed with another, progeny will expect to be have 3:1 ratio. So here is the cross of dihybrid cross. You have gametes of capital G, small t, small t. So this is green color, green pod, and tallness and dwarfness. So there is a tallness and dwarfness and color of the pod. So both green podded tall, yellow podded dwarf. Yeah, so you have F1 generation. So in F1 generation, you have all green and all tall. If we cross between yellow podded dwarf and green podded tall, that is capital GT and small gt after getting the gametes out of them. So you will have all your F1 generation to be tall, green, podded tall. And then you further cross them, you will have respectively these different generations in this dihybrid cross. Now, back crossing is a crossing of hybrid with one of the parents. So you cross with the parents itself, individual, and in order to have offspring with a genetic identity which is closer to the parents. So if you want to have genetic identical things of the parents, you cross them with the parents itself to have the same characteristics of your offsprings. So that is known as offspring or back cross. Test cross, what is test cross? It's the same or allele so dominant over others. The phenotype of organism does not always reflect its genotype. So a recessive phenotype is only expressed with organism is homozygous recessive. So a pea plant with green pods may be either homozygous or heterozygous. So to check whether you have homozygous or heterozygous, that's why we do the test cross. So you have a dominant phenotype, but we don't know whether it is a capital P, capital P, small p, the purple color. And but we have recessive phenotype in which we are aware of that it is small p genotype. So if capital P and capital P small p, if offsprings, if you cross them, so if everything is capital P it came out to be capital P and small p, then all offsprings are capital P capital P. If your offsprings are purple, they're all coming out to be purple, so that means it was capital P capital P. But if your half offsprings are capital P purple and half to be white, that means that was recessive. So this is heterozygous, capital P and small p. So by this test cross, by multiplying with another genotype which is known, you can tell to the another phenotype what could be the phenotype of that. So that's it from this type. So maybe your questions. But YY mouse is known to be lethal. If the capital is present, capital Y, so most likely your mouse will die. But if it is, that's what the gene is known for. That's what's the duty of that gene in specifically in the mice. If the capital Y is, if they are present in the dominant nature, which is known to be lethal, whenever that gene is present, the mouse has to die. But if they are present in the recessive form or in the heterozygous form, mouse will have either yellow color or non-yellow color respectively. So this is the trait of the allele actually. So that's why they are known as lethal genes. So the genes which cause the death of its carriers when in homozygous conditions, so they were present in the homozygous conditions, are called as lethal genes. That is, they are both in the capital form or small form also. So Mendel findings were based on the equal survival of genotypes in normal segregation. 3:1 is modified into 2:1. So most of the time we saw 3:1 ratio, but now we saw a ratio of 2:1. That is, yeah, two were surviving, one was dying. So lethal genes have been reported in both animals as well in plants. In mice allele, yellow coat color is dominant over the gray. When the cross is made between yellow and gray ratio, 1:1 ratio was observed between mice. And this indicates that yellow mice are always heterozygous because yellow homozygotes never born because of the homozygote lethality. And such genes, these things were not observed by Mendel, so he never studied them. So he always give ratio of 3:1, never 2:1. So that was something new for the Mendel also. So in these are the in the above so these in the above mentioned, it's not always that dominant will be present. It's also the non-yellow will be also that is the recessive part could be also be lethal, not the dominant always. So this depend upon from one generation to another. I hope it's clear now. Synthetic lethality, that's a good question. Let's Google it. Uh, lethality arises when a combination of deficiencies in the expression of two or more genes lead to cell death, whereas deficiency is only one of these genes does not. So these deficiencies could be through mutations, epigenetic alterations, or inhibitors of one of the genes. Okay. So this is an example of synthetic lethality. This is a normal cell and gene A, gene B are present. So they produce single mutant and overexpression. So here these are viable genes present. So this is viable, but double mutants, when they make double mutant out of them. So gene A and gene B both have with a star expression. So this is double mutant, and here the gene B is removed, and there's inhibitor also, which is not letting the gene B to be activated, and then there's overexpression of gene A. So in these specific conditions, so this is synthetic lethality. So we are producing them by our own case. So by producing double mutants, by not expressing one gene and letting the other gene things to be overmasked by others. So these are comes out to be um, synthetic lethality, that by stopping one expression on our own, oops, we can we lead to this. So yeah, it could die, as in, so in that case, that's why they say that it's sometimes recessive could also die. But in that case, specifically when they're doing this cross of the yellow coat and the gray coat, white coat, so in that case, mostly capital Y will die, not the small y, because they are in recessive. And also I discussed in the start of this lecture that how the expression is governed. It's the active polypeptides. So in that case, its active polypeptides are producing the functional enzymes, whereas in the recessive genes they are producing incomplete non-functional polypeptides. So in that case, capital V, Y. So we were seeing some functional enzymes being produced, which is leading to the death of the organisms. Whereas the small y doesn't produce any lethal genes and let the mouse be survived. Yeah. Now comes the next part. Types of gene actions. So this is the next part. So here we will discuss various other exceptions to the Mendel's law, like epistasis, masking of one gene to another gene, and seeing many things for. Before we go ahead, let's take some break now here, so that we can we can start with a fresh start. So let's take some break here. So it's almost 46 now, 47. So we meet at 55 then. At 55 we start our next topic. So just grab a cup of coffee, or I will grab one for myself, and then we meet us at so we have some break now. for 5 minutes. So we meet at 55 then. Okay. So and we start with next topic, that is types of gene actions. So, next topic, types of gene action. So, are you all back? Shall we start? So, this is the second topic from Unit 1. So, here we are dealing with the intrallelic and interallelic interactions. So, if the interaction is done of single character, maybe that is dominant or incomplete dominant, are called intraallelic interactions. But once there is interaction between different pairs of alleles, so different present on the different chromosomes or different places, influencing a character of an individual is said to be interallelic interactions or epistatic. So gene that has masking effect is called, the gene which is masking the other gene is called epistatic gene. So that if there is one gene A, so this is gene A and this is gene B. So this is if it is masking the effect of gene B, and that gene will be called epistatic gene, and whose effect is masked, that is gene B, that will be known as hypostatic gene. So that's the difference between epistatic gene and hypostatic genes. So epistasis leads to modifications of normal hybrid or trihybrid segregations in the F2 generations further. So term epistasis was coined by Bateson in 1909, and various types of epistatic gene interactions were discovered. So like recessive epistasis, dominant epistasis, dominant and recessive epistasis, duplicate recessive epistasis, duplicate dominant epistasis, polymeric gene interactions. So all they came to be part of various types of gene of actions. So which are giving respective ratios: 9 is to 3 is to 4, 12 is to 3 is to 1, 13 is to 3, 13 is to 3, 9 is to 7, 15 is to 1. So all exceptional things that they found out. So duplicate recessive epistasis. So students, please open your minds and try to understand each topics in upcoming time in more detail. So each thing is quite important for us now. So duplicate, just remember the definitions, how definition itself saying everything. Duplicate recessive epistasis. So whenever the recessive alleles like small letters or at either of the two loci can mask the expression of the dominant genes. So recessive genes which are present, so like let's say small a and small b, So they are like they're masking the effect of capital N small b, capital N capital B. So the dominant genes, that is called as duplicate recessive epistasis, or it's also known as complementary epistasis. So best example is flower color in sweet pea. So flower color in sweet pea is the best example for this. So in this example, purple flower of sweet pea is governed by two dominant genes, that is capital A and capital P. And when these genes are separate individuals, capital A small b, small a capital B, and white, and they produce white flowers. So whenever they're in this form, small, they produce white flowers. So cross between capital A and capital B and white flower, these strains produce purple flowers. So first, whenever we have a cross of purple, capital A capital B, and small a small b, so we have F1 generation to be purple. But once we segregates their genes, so capital A capital B, capital A small b, small a capital B, small a small b, we made a cross between each other in the F1 generation, in the F2 generations. So what will happen? We will see a lot of white genes to be present. So why they are present? Because whenever they are present in two forms, small a small a, small a small a, capital small b, so they will be white. So they will epistatically will govern the results of that. Like here we have small b again present, and we have here small b present, we have small a present, small a. So by this case they will they will dominantly act as a dominant to be epistatic to it. So in this case F1 produce purple and white flower in the ratio of 9:7 ratio, not into 9:3:1, but we have a 9:7 ratio. So 9 are purple flowers, 7s are white flowers. And here the recessive gene A, small a, is epistatic to capital B or small b and masks the expression of the genes. Another small b is epistatic to capital N small a and masks the expressions. That is how the epistatic thing works out. So that was about complementary. First was the duplicate recessive epistasis. Now comes the duplicate gene actions. Uh, when a dominant alleles, either of the loci, can mask the expression of recessive alleles, so in this case dominant gene is masking the expression of the recessive allele. So it's the another way around from the previous sections. It is known as duplicate dominant epistasis. So dominant genes are there taking care of the recessive genes. So in the Ross on character, so this is on characters, that length of your rice, that is known as your on. This is, we can see on, and this is onless without seeds here. So there are two types of characteristics. On is dominant and onless is recessive. Okay, and dominant genes are present by capital A and capital B, and onless rice is presented by small b. Your first generations came out to be capital A and small a, capital B, small b. That is on rice. You have first on rice, and the onless condition will be always be with the homozygous recessive state, that is small a, small b, small b. So you will have mostly in that case, if either case, so a cross between on and onless was done, and on plants were produced in F1 generation. Then F1 generation produced on and onless versus onless into ratio of 15:1. So 15 were on and 1 was onless in the F2 generations. Why? Because A is epistatic to small a and small b, and B is epistatic to small a and small b. So these individuals will always produce on characters. So whenever capital A or capital B's are present, so like in all these cases, in all these cases, you have your on characters are present, but in the rest, in the small case, only in this case you have small a, small b, to be present. Then comes the inhibitory gene action. So in this type of epistasis, a dominant allele at one locus, can mask the expression of both dominant recessive genes at the second locus. So one dominant gene could mask the function of your dominant as well as recessive genes at the second locus, and this is known as inhibitory gene interaction. An example of this is anthocyanin pigmentation in rice. So anthocyanin is one example here, which has a pigmentation of anthocyanin in the rice. So the green color of plant is governed by gene I, which is dominant, okay, over the purple color. The green color is dominant, gene I, over the purple color, and purple color is dominated by the gene capital P. So when a cross is made between capital I, capital small p, small I, and capital P colored plants, F1 was green. So you have first plant to be green, and intermating with F1 plants producing green purple plants into ratio of 13:3. So here you have a ratio of 13:3 with the three purple colors plants like these three, in which you have mainly the purple color to be dominant if they are present in capital P, and yeah, and rest is all green. Rest is your all green plants are present. Then comes the recessive epistasis, supplementary gene actions. Here one dominant gene has its own phenotypic effect, and the other dominant gene has its no effect. So dominant gene will have its own phenotypic effect, another dominant gene will have no effects of its own presence with the first genes. So thus, in supplementary gene action, dominant gene of one gene is necessary for the development of the concerned phenotype, while other gene modifies expression of the first gene. So both are interdependent of each other in that case. So you have purple RRPP, and you have small r small p red. Then you have capital R small r, capital P small p purple, and then you segregate them into different parts with RP, 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 and RP, and then you have these ratio of 9:3:4 ratio of purple, red, white according to the supplementary gene reaction. Then comes the additive factors. So in this case, two genes they come together and their genes are affected. They are they come together and their things plays role together. So they have additive effect of the things. So if the two genes controlling a character produce identical phenotype, when they are alone, that is with homozygous recessive condition of the other gene. But when both genes are present together, the phenotypic effect is enhanced. So both genes are present, the effects get enhanced, and it's known as cumulative or additive effect, and that is known as your complete dominance here. So in barley, two completely dominant genes, gene A and B, affect the length of the ons. So length A and B, they are present, they affect the length of the ons, and thin needle-like extension of lemma genes, capital and small b alone also. So if they are present in this form, like small a and small b, if they are not present fully, so they give medium length to it. A is the same as that of B. So what we can see is that if the both are present and we have long, both capital and capital B are present, we see long. But if there is one is small, so one is not present as a big dominant, so we see medium ond. So we see medium ond like length L. But only in one case we have onless, in the last case. The rest is long or ond or onless. So like this. We have three different ratios because of the additive effect, additive factor effect. Then comes the dominant epistasis. This is an example of dominant epistasis found in the fruit color, that is white, yellow, green. So white color is controlled by gene W, yellow color by G. So white is codominant over the both yellow and green fruits are produced in the recessive conditions WWGG. Then we made a cross of like this, like here we can see white fruits WWGG and yellow fruits small w capital G. So cross was done between white and yellow fruits, produce F1 plants to be white fruits. So you have F1 plants to be white fruit like WD capital W small w capital G small g. Then you segregate these genes again, so you have different ratios, and according to the dominant epistatic things we have three ratios, that is white, yellow, green. So if they're present in the recessive form, so we have only one green, but if they are present in this form, capital G is present once or twice, so we have yellow. In the rest of the cases, it will be always because of the W is present in the capital in most of the cases, so it's always white. So that is how dominant epistasis work in this case. So this is also reported in the skin color in mice, seed coat color in the barley in that case with the dominant epistasis. So we are done with all types of actions, gene actions. Now comes some modifying genes. So here we are discussing about some modifying genes from the point of the mouse color coating, the coats they have. So they are group of genes which enhances or reduce the phenotypic effect of major genes. Such genes have small or cumulative effect on the expression of major genes. As a result of continuous variations is generated in the phenotypic governed by a single major genes, which converts qualitative character into quantitative one. In rats, guinea pigs, rabbits, piebald spotting is produced by recessive genes which present in the homozygous state. So whenever in these mouse, these spots that you're seeing, this is called piebald, and these are present only when they are present in the homozygous state, that is small s, and the degree of spotting depends upon the modifying factor designs S1, S2, S3, which enhances or reduces expressing these spotting genes with cumulative on spotting. So this is how you see uh, cumulative genes present as S1, S2, S3, how much they will be present, the more the balding or spotting will be present in that. So most quantitative characteristics of crop may be determined in similar fashion. Some modifying genes affect more than one character. So we have more than one character being affected because of this. So in piebald spotting, the modifying factors produce some spotting even in the absence of spotting genes, but the effect is much more pronounced in the presence of small s. So the effect is, as I discussed only, so how much the small s is present, only the effects will make the effect of your piebalding. So spotting gene S is a major gene controlling the spotting, while modifying genes or minor genes affecting this trait. So they are major genes and minor genes. Major genes are controlling the spotting, whereas the minor genes are affecting this trait, like how much present, how much it will be present, how much it will be not present. So now inheritance of quantitative characters. So they are color concept of polygenes like color, sex, which shows distinct categories, are known as qualitative characters. They are usually governed by one or major genes of oligogenes. Characters like length of ear in corn, yield of grain, yield of milk, stature, they all comes under the and governed by the minor genes, so multiple genes or polygenes. So characteristics features of quantitative characteristics is continuous variations and marked influence of environment on the expressions. And then there is a transgressive segregations. So this is transgressive segregations appearance in the F2 individuals with the higher or lower intensity of characters than the parent is a transgressive segregations. Then comes some different terms like penetrance, polydactyl, and expressivity. So these terms come sometimes in your exams. So frequency with which gene produce a phenotype or visible effect that we are seeing is known as penetrance. Or penetrance refers to as proportion of individual which exhibit phenotypic effect of specific genes carried by them. And whereas polydactyl is a condition with the extra finger, like Hrithik Roshan has extra thumb, I guess, or toe in a man is due to the presence of dominant gene P. The normal condition is produced by the genotype capital P, and the genotype and small p produce polydactyl. Some heterozygous individuals are not polydactyl, so capital P small p, therefore the genes has penetrance of less than 100% and said to be incompletely penetrant. A gene through penetrant may be quite variable in its expressions, and degree of expression produced by penetrant genotype is termed as expressivity. So degree of phenotype expression, so degree of whatever the phenotype that you're expressing, how much they are being present of a penetrant gene is called expressivity. So ability of gene to produce identical phenotypes in all the individuals carrying its individuals is known as incomplete expressivity. So that's it for today, students. So this qualitative and quantitative traits, we do it for tomorrow. So it's not good we do so many things in one day. So we do it with a very nice pace, and we will do it tomorrow, this thing. So I'm thinking of skipping this today, and let's take your questions. So bache Nishtha, so qualitative, quantitative, we will go in very detailed manner about them, so don't worry much about it. Some Malai, sorry. Um, Minor genes. So bache, there are major genes and minor genes. Mm, so in this case, how much character, like whether the spotting is present or not, that is dependent upon the gene S, and its effect like S1, S2, S3. These are the minor genes, so they will be modifying genes, and how much they should be present, their trait should be present, that will depend upon its minor character by the minor gene. So the degree of effect is done by minor genes, and whether that gene will be present or not, that is done by the major genes. Yeah. So for today, I will share you all the things. This is for today's lecture. Types of gene action. Okay, so today I hope everything is cleared with you, and see you tomorrow, same time, same place, with lot of enthusiasm and a lot of awesome spirits. And yeah, wish you all good luck. Have a nice Tuesday. Wish you all the best for your upcoming endeavors. Thank you so much. Take care. Bye bye. Good night. I will end the stream now. I have my next lecture of MBBT. So I just have to prepare some for that too. Okay, bye.